Hello students, welcome to the academia. Today our topic is lipid, and lipid is one of the most important compound of our body, and lipids are the broad category of organic compounds. So what are the organic compounds? Basically, organic compounds means those having the carbon in their structure. So the phospholipids are one of these heterogeneous compounds included in the category of biological molecule. So the phospholipids basically they are the heterogeneous compounds. So these are the heterogeneous compounds, and phospholipids are characterized by having a phosphate group in their structure. So basically these lipids having a phosphate group in their structure. Okay. So due to having this phosphate group, they become polar. They are the most prominent lipids present in all type of membranes. So generally we know in the cell membrane, our human body, the unit of the body is cell. So the cell having the membrane, and the membrane is made up of basically the amount of lipid is highest, then the proteins, and then the carbohydrates. In the membrane lipids, the amount of phospholipid is maximum. So the phospholipid is one of the most important lipid of cell membrane. So if you recall the structure of the cell membrane, you can remember this. These kind of lipid structures were there, and they form the double layer. This is called the bilayer, within which the proteins are embedded. So these kind of structures were present in the membrane. So these membrane lipids are basically phospholipids. Other lipids are also included, but the phospholipids are most important. So next we found the general structure. Phospholipids include a large variety of compounds that have different structures and properties. So moving to the structure. However, it must, for a phospholipid to have two fatty acids attached to alcohol via an ester bond, and a phosphate group, as I have mentioned earlier, attached to the same alcohol molecule via a phosphodiester bond. So what they need? It must have two fatty acids. And Okay, so these things are must. The presence of phosphate group in phospholipid make them polar. As I have already mentioned that this phosphate group make the phospholipid polar. However, the fatty acids impart the hydrophobic characteristics to these compounds. Thus, each phospholipid has both polar and non-polar ends. Generally, what we know that the lipid molecules are hydrophobic in nature. And what does it mean? Hydrophobic means hydro means water, phobic means phobia. That is those substances which don't attract water, they repel water. Hydrophobic means repel water. So the phospholipid molecule also, such lipid which is hydrophobic, that is some portion of the lipid is polar but some portion is also non-polar. So due to these characteristics they become hydrophobic. So like the other lipids, the phospholipid also have polar head made by phosphate group and tails, non-polar tails made by two fatty acid present in them. So can draw the diagram like this, like if it is the phospholipid molecule. So this is the phosphate group, having phosphate group which is polar, and these are the non-polar tails. So this is head which is polar. Okay, so this is the actual structure. The hydrophobic tails are attached to the polar head of the compound. So in the structure we can see that the head and tails are attached. One is polar, one is non-polar. Now in the diagram can see the structure of individual phospholipid, and here the hydrophobic head and tail, and the formula has given. So can easily understand that how it is formed. So the formula is important. Now moving to the classification. Phospholipids are divided into two broad categories based on the alcohol present on their backbone, and these are glycerophospholipids and sphingophospholipids. Both of them are present in the cellular membrane and perform important functions. So, number 1, glycerophospholipids. So these are the phospholipids which contain glycerol as an alcohol in their backbone, and they are also known as phosphoglycerides. So it is important, they are also known as phosphoglycerides, and they are the most abundant lipid in the membrane of the living organism. So that they are the most abundant lipid, glycerophospholipids, and they contain either a molecule of phosphatidic acid or are its derivatives. Next we move and see that what is phosphatidic acid and the components, by which components the phosphatidic acid is made up of. So a molecule of phosphatidic acid is made up of two components. Number 1 is diacylglycerol. I think many of you know this. That is DAG, and another is a phosphate group. So the diacyl glycerol contains glycerol alcohol along with two fatty acids attached to the first and second carbon of glycerol via ester bonds. The addition of a phosphate group to the third carbon of glycerol via phosphodiester bond converts DAG into phosphatidic acid. So with the DAG, the phosphate group is attached to form this phosphatidic acid. Having understood the structure of the phosphatidic acid, it will easier for us to have a look at the structure of various glycerophospholipids. So this is the way you can see that from DAG how the PA, that is the phosphatidic acid, has made. So this is the way. So next, after phosphatidic acid, in the glycerophospholipids there are some phosphatidyl alcohol. So what is phosphatidyl alcohol? So many glycerophospholipids simply contain an alcohol esterified to the phosphate group attached to the third carbon of the glycerol. And these phospholipids are given the names by adding the names of alcohol after the prefix, and it is called phosphatidyl alcohol. So examples of such lipids are, so examples include phosphatidyl serine. I think many of you have know this. It contains serine alcohol attached to phosphatidic acid. Can you understand? So phosphatidic acid we see earlier. So if the serine is attached to it, then it is called phosphatidyl serine. Next, phosphatidyl ethanolamine. Another example is phosphatidyl ethanolamine. So it has ethanolamine attached to phosphatidic acid. So phosphatidic acid means PA. Next, phosphatidyl choline. In this phospholipid, the alcohol is choline. So here the alcohol is choline. So choline, serine, ethanolamine. So these are the alcohol part. Okay. And next, phosphatidyl inositol. Phosphatidyl inositol. I am giving the commas. So inositol is the alcohol in this phospholipid. So here inositol is the alcohol part. Next, phosphatidyl glycerol. Phosphatidyl glycerol. Here the glycerol attached to the phosphate group. Okay. So these alcohol molecules are added and it forms the phosphatidyl alcohol. After that we move to other derivatives like cardiolipin, plasmalogens, etc. Now another derivative is cardiolipin. So it has two molecules of phosphatidic acid attached to an additional molecule of glycerol, which is an alcohol. So the phosphate group of two phosphatidic acid, so these are the phosphatidic acid and the phosphate groups groups of 2 phosphatidic acid make ester bond with glycerol, resulting in the formation of diphosphatidyl glycerol, also called cardiolipin. So actually the phosphate groups of the 2 molecules of phosphatidic acid, they actually make ester bond. So they make ester bond with what? With glycerol, and resulting the name of the molecule is diphosphatidyl glycerol. The molecule name is diphosphatidyl glycerol or cardiolipin. So it is called cardiolipin. Okay. So next one is the plasmalogens. This is another derivative. So they don't have fatty acid. Plasmalogens have an alkyl group, okay, attached to the first carbon of glycerol, which is an alcohol. The alkyl group is attached via an ether linkage to the glycerol, and the rest of the structure same as phosphatidyl alcohol. Okay, so why they are given these names? The plasmalogen are given names by adding the name of alcohol after the prefix phosphatidyl, such as phosphatidyl choline. So can see the spelling, it is phosphatidyl choline. So plasmalogens are also known as phosphatidyl choline, but it is not similar to phosphatidyl choline. Okay, so keep this thing in mind. So next one is the platelet activating factor. So it is another derivative of phosphatidic acid, and here the first carbon of glycerol has an alkyl group, while second carbon of acid has acetyl group, and both the groups, that is alkyl group and acetyl group, both the alkyl and acetyl groups, so how they are attached? They are attached by the ether linkage. So here there is ether linkage. So the linkage is ether linkage. Because of the presence of ether linkages, plasmalogens and platelet activating factors are collectively called ether glycerophospholipids. So the platelet activating factor and before that we have seen plasmalogen, both are called ether glycerophospholipids. So their name is ether glycerophospholipids. Okay, so both plasmalogen and platelet activating factors both are called this. Next, if we move to the synthesis part, so the number one classification of the phospholipid was glycerophospholipid. So in our next video I will discuss about the sphingophospholipids. So the glycerophospholipids, the synthesis occur in two ways: either transfer of phosphatidic acid to an alcohol or transfer of phosphomonester of alcohol to diacyl glycerol. So this is all about the classification of glycerophospholipids, and next in the next video I will go through the sphingophospholipids. So if you have any query question, you can ask or can comment in the comment box. Thank you.